UCC, and I'm doing research on biological nanopores for DNA sequencing in the electrical and computer engineering department. My mentor is Danielle Guerra, and my PI is Luke Theograjan, and we're funded by the National Institutes of Health.
It's on. You got it. We'll start. Which is engineering, you can also just do that. Maybe.
Okay. So, conventionally, DNA sequencing is done using biological machines. So, there are specific enzymes that evolve to recognize a DNA sequence and synthesize a complementary DNA strand. And that's what DNA polymerase does here. This enzyme is used in a process called Sanger's method, which actually received a Nobel Prize in chemistry in 1980. But as you can see, it's a highly technical process that involves chemically labeling the DNA and modifying it. And so it takes a lot of time. You can only sequence 9,000 bases in 24 hours. And so there's a better way to do this. And in comes nanopore technology. Nanopores, as the name suggests, is a pore with a diameter about a nanometer, or a billionth of a meter, about the width of DNA itself, actually. So why use nanopores for DNA sequencing? Well, first off, it's highly efficient, so you can sequence potentially really long DNA strands compared to the Sanger's method. And it's affordable. So about a decade ago, it cost $100 million to sequence the human genome, the 3 billion base pairs that make it up. And currently, it costs somewhere around a few $1,000s to sequence it. And so we think nanopore technology will continue this trend and make it even like $100 or less. It's extremely useful to know the DNA base order, to identify genes and see how they're associated with diseases like cancer, diabetes, immune disorders. You can also identify mutated genes by comparing it to a reference genome that you sequenced. And so really this ushers in this whole era of personalized medicine where you can tailor individual drug therapies for an individual's genetic makeup. The technology may go from the lab and then soon into the industry when it becomes commercially viable, and then to the clinic where sequencing could become maybe as commonplace as, say, getting your blood pressure taken, and then even to the household, like in this picture here, where you could plug in your sequencer to your computer and like a flash drive and read out your results there, as opposed to the current technology, which involves these bulky lab machines that require technicians and it's super expensive. This research involves many disciplines, from biochemistry to biophysics to computer science to mathematics, all of which play a huge role in the final design. So how does it work? So what you do is you take a chamber and you fill it with water, salty water, and you apply voltage. And you divide it so you have the pore in the middle, and you apply voltage. And the voltage will drive a current, a flow of ions through the pore. And then you take your DNA and you throw it in there. Since DNA is negatively charged, it'll be repelled by the negative electrode and move towards the positive electrode. And it'll actually go through the pore. And as it goes through the pore, it'll create little diminishes in current. And that is what you read as your signal for DNA sequencing. And the beautiful part is it's actually sensitive to the individual bases of DNA as it moves through the pore. So this is what the setup would look like if you enlarge there. You would have a protein pore embedded into a polymer membrane platform. And that's specifically what I'm researching on this summer, the platform for the nanopore. So there's several advantages to using polymers. They're highly stable, and that's good news for the protein that you're going to insert. And it's long-lasting, so it's actually commercially viable, unlike the lipid membrane, which lasts from a few minutes to a few hours if you're lucky. And they're extremely fragile. I hate working with them. So unfortunately, though, we can't just insert our protein into our polymer membrane and not worry, because proteins behave differently in different bilayer environments. And the polymer membrane is not the protein's natural environment, the lipid is. And so when we insert it, we'll have what's called hydrophobic mismatch. It's where the length of the protein differs from the thickness of the bilayer. And this is super energetically unfavorable. So the bilayer likes to deform around the protein to compensate. And that in turn creates a conformational change in the protein. And so the extent of this deformation depends heavily on properties like thickness, elasticity, curvature, and thus bilayer properties can give rise to certain protein conformations. And when you have different protein shapes that are possible, you also have different protein functions that are possible. So we really need to understand our polymer before we can start inserting proteins. And our solution is to use Gramicidin A as a molecular force probe. Gramicidin A is a polypeptide that we can insert into our polymer, where it'll exist in two forms. The non-channel form here has these two subunits, and then the channel form, those two hook up and form what's called a dimer, which will conduct ions. And when it does this, it'll create a deformation just like in the previous slide. And it'll exert what's called a disjoining force on the dimer, trying to break it up into this non-channel form again. And that disjoining force is related to properties like the properties that I discussed earlier. And so by measuring the lifetime of the dimer, we can deduce certain quantitative properties that allow us to understand the energetics behind our polymer. So firstly though, before we do that, we have to actually make membranes. And we do that especially with this Teflon chamber here. The membrane forms in this 50 micrometer hole in the middle of this thin sheet of Teflon that we vacuum grease onto the chamber. And when that's all assembled and sealed, we'll take it over to the Faraday cage, where we'll hook up the electrodes and pour in the electrolyte solution of the salty water, and we'll apply voltage. And the voltage, and if all goes well, we'll form stable membranes that we can track in real time using the camera on the bottom that's hooked up to our PC. And our amplifier that we use is sensitive to extremely small current levels in the picoamp range. So that's a potential problem. We can get external electric fields interacting with our experiment. So once we form a stable membrane, we can pipette Gramicidin A near the membrane and just look for characteristic current traces that are indicative of it associating with our bilayer. And then post-experimental processing, we can construct graphs like this, where we can see single channel insertions and also judge their lifetimes from that. So here are two membranes we formed. This is a membrane formed with a 100 micrometer aperture. Those membranes are usually less stable than the 50 micrometer apertures here, which I usually work with. There are some, okay, so actually, the rough textured regions you see that kind of look like the surface of the moon here are a mixture of polymer and solvent that we paint onto the Teflon prior to applying the voltage. And that sort of acts like a primer before we start forming it. And then there are some physical indicators we can use to see stability. One is the sort of outer wider rim of solvent that forms right when it becomes stable, and then as you go inward, it gets more darker. This will actually start expanding and take up the entire aperture. So here you can tell this is like an initially formed membrane, and this one's a later, more stable membrane. And we can also judge its stability electronically using capacitance, and we do that by modeling the membrane as a parallel plate capacitor. Parallel plate capacitor is basically when you have two conductive regions separated by a non-conductive insulator called a dielectric. And we model our membranes bilayer or hydrophobic region as the dielectric. And then when we do that, we can apply the formula capacitance equals epsilon A over D, where epsilon is the permittivity of the polymer, which is a known value. And then A is the area of the membrane, and then D is the thickness. And so you can see the capacitance is related inversely to the thickness. And so when we first form membranes, initially there's like a glob of solvent right in the bilayer. And as it thins out and gets more stable, it'll actually attach to the Teflon aperture and form like a kind of bridge between the Teflon and the bilayer. And the thickness will decrease. So one way we can see stability is that over time, the capacitance will increase as we form our membranes. Here's protein insertion data at 180 millivolts. This is actually not Gramicidin A. This is the protein pore you saw earlier, Mycobacterium sphygmatis porin A. You can see the stepwise conductance increase is characteristic of insertion. We expect Gramicidin to have a similar increase in conductance, but at a smaller level because it's a much smaller polypeptide. And we're still working with Gramicidin. We had some problems, like we tried to insert it into a lipid membrane and it ended up crashing out of solution. So we need to keep refining the protocol and methods for our Gramicidin assay, and that's coming up. But I have learned how to form membranes and the whole process leading up to it, and it ended up being really difficult to learn the methods involved in that. Future research is continue with Gramicidin and hopefully to understand our polymer so we can get stable protein insertions. And then long term, as a group, we want to construct a nanopore array, which would allow high throughput sequencing, which basically just means we could parallelize the sequencing and have multiple cores that are doing it simultaneously. And thank you.